Hey guys, my dear beloved student, today we will be discuss about digestion and absorption of protein. So let's make it really simplified and amazing for you. First of all, I love to draw a very delicious diagram for you. Just you consider that is a leg piece of chicken, right? Wow, that is a really beloved diagram for you. That is golden fry leg piece of chicken. That's mean it is meat, meat, right? That is for my non-veg student, and it makes sure I make another diagram for vegetarian, my vegetarian student. That is actually a bowl of pulses, dal, right? Dal, DAL. That means pulses, and pulses contain, as well as this meat contain lots of protein inside that, right? This food is really rich in protein, right? Protein. And some another diagram for the source of protein is, that is the egg, right? That is the egg. And all this food is really rich in protein, right? And today's topic main agenda is that how this protein is digest inside our digestive system, and after the digestion how this protein is absorbed inside our intestine, right? That is the main agenda for today's topic. Wow, right. And you know very well, my dear student, you know very well that this protein is actually a complex structure, right? Complex, PLEX, complex food, right? This protein is really complex food before digestion, right? But after digestion of this protein is really convert into simplified food, that is split into amino acid. I shortly called AA, just I mentioned amino acid, right? Wow. And I make a diagram for you, just a basic diagram of amino acid structure. And amino acid contain on amino group, that is NH2, and carbon, again carbon contain a alone hydrogen, and again there is a alkyl group that is represent as R, as well as a carboxyl group which is actually COOH. That's mean that is the main basic skeleton of amino acid, right? Molecular skeleton of amino acid. And amino acid contain, you can see in this diagram, amino acid contain on amino group, that is amino group, and that is alkyl group, and that is your carboxyl group, carboxyl group. And that is the basic structure and skeleton of amino acid. Ultimately, after the digestion of protein, that's mean after the digestion of this complex food protein, it simply convert into simple structure, that is called amino acid. And this amino acid is actually absorbable inside our absorbable inside our beloved small intestine, right? Small intestine. But how this event occur, we'll learn this topic step by step, right? And that is the sum of example of The source of protein actually. That's mean high protein. That's another high proteinaceous example I missed it, and you will tell me what is this diagram name, right? Wow. Very good. That is, you can easily point out that that is a fish, 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 right? And also meat, pulses, egg, and fish contain lots of protein, right? And this protein, when we consume this protein after the processing, then this protein is really convert into amino acid by the action of protein digestive enzyme, which is inside our stomach, and as well as not only stomach, it is actually in our intestine, is in our GIT, gastrointestinal tract. And more information about you, that this fish contain most easily digestible protein. Wow, this fish contain most easily digestible protein, right? That is MCQ, or that is short question point of view. It is it have very great importance, right? Proteins. Wow. That is, now we have to focus about the basic design of our digestive system. And first of all, we have to discuss about digestion of protein, and after that absorption of protein. First of all, you have to copy this board, right? Now we have to draw a simplified diagram for you, right? And that is, you know very well, that is the diagram of a very pretty and beautiful face. And face contain actually that is mouth, right? This portion is mouth, MOUTH, mouth. No need to mention that, you already know that. And mouth is bounded by two jaw, right? Upper jaw as well as lower jaw, right? That is your lower jaw. And upper jaw is called maxilla, MAXILLA, maxilla. And lower jaw is called mandible, right? And you only can move your mandible, not maxilla. We cannot move our maxilla, right? We cannot move our upper jaw, and both of these jaw contain teeth, right? Overall, it's make a very funny diagram. Wow. That is teeth, and both of jaw contain teeth, and in between them there is your greedy tongue, right? Okay, that is your tongue. Now when you see some delicious dish, right, just like proteinaceous food, that is meat, dal, and lots of egg, right? Egg which is boiled, or egg curry, and its smell is really pretty, right? The smell is really pretty, right? That is smell, and the smell is just accepted by your nose, and then salivation is start by seeing this pretty and delicious meat and egg, right? MEAT, right? Meat. And you wish to eat them voraciously, very actively, and you are really very humble to eat this food. Wow. And after eating this food, this proteinaceous food is first breakdown by physically, right? This food is digest physically by the help of teeth, tongue, and jaw. Right? Jaw is bounded by mandible as well as maxilla. So first MCQ or first short question for you. Protein is first of all physically digest by teeth, tongue, and jaw. And on more MCQ for you, on more question for you is that there is no chemical digestion occur inside the mouth. There is no chemical digestion of protein. No chemical digestion. No chemical digestion. No chemical digestion of protein inside the mouth. And on more important question, that carbohydrate digestive enzyme is present in our saliva, but our saliva have no protein digestive enzyme, right? And after that I just simply draw esophagus there. Just simply draw esophagus there. There is the exact, not the exact position of esophagus, just I draw a hypothetical diagram for you, right? That is esophagus, and after that there is your hungry stomach, right? Hungry stomach, pouch like organ, and it is the temporary, actually act as temporary reservoir for food, right? And how many time food can reserve inside the stomach? It is exactly near about 4 hour, HOUR. That's why we feel hungry after 4 hour, right? Because stomach maximum store the food up to 4 to 5 hour, right? 4 to 5 hours. After physical digestion of protein, this protein is enter into the stomach, right? This protein is simply enter into the stomach, right? And after stomach, there is presence of C-shaped curve, and this C-shaped curve portion is actually called duodenum of small intestine. DUODENUM, right? Duodenum. And it is make a C-shaped curve, alphabet like C, right? Now your protein, targeted protein is now inside the stomach, and this protein is first of all physically breakdown by teeth, tongue, and jaw. This protein is just physically digest by your teeth, tongue, and jaw. Your teeth, tongue, and jaw chop this protein into the small piece only, right? There is no chemical digestion because our mouth, our saliva don't have any protein digestive enzyme as well, right? Now real function, real digestion of protein is start from the beloved stomach, right? And you know very well, this stomach is responsible for releasing of HCl, hydrochloric acid, as well as it is also responsible for production of protein digestive enzyme that is called actually pepsinogen. What is the enzyme for digestion of protein? It is pepsinogen. Pepsinogen, right? And this pepsinogen is exactly not a functional enzyme. This pepsinogen now is inactive enzyme, right? This pepsinogen is really inactive enzyme. Pepsinogen, right? Pepsinogen is inactive enzyme. When this pepsinogen is come contact with the HCl, right? HCl. When this pepsinogen is come contact with HCl, and then this pepsinogen is convert into active protein. This pepsinogen is really convert into what? Active protein, that is called pepsin, right? And really this pepsin is now very happy. Because of why it is very happy? Because now this pepsin can work on the digestion process of protein, and it is really a very good digestive enzyme for protein, right? And this small fragment of protein which is physically digested by teeth, tongue and jaw, they are again digested by this pepsin, and this protein is fragmented into small and smaller pieces, that is called small polypeptide and polypeptide as well, right? This protein is under the function of this actually pepsinogen. This protein is further digested by chemically digested mention. You have to focus on this point. Protein, first time protein is digested chemically by the action of enzyme pepsin, and pepsin is not directly secreted as a activated enzyme. It is fast released as a inactive enzyme, that is called pepsinogen, and pepsinogen when come in contact with the HCl, which is also secreted by the stomach, and when this HCl is come contact with this pepsinogen, then this pepsinogen is really convert into active pepsin, and this pepsin can digest the protein further, right? And this protein now fragmented digest into small fragment or small pieces, that is called small polypeptide and polypeptide as well, right? That is the function of pepsin. And one more MCQ, one more short question for you. Inside which cell of our stomach is actually responsible for releasing of pepsin? Can you tell me, my dear student? Which cell of stomach contain lots of cell, right? Stomach contain lots of different type of cell, and my question is that which cell of stomach is responsible for production of pepsin? The beloved cell which is responsible for production of pepsin is called chip cell, right? Chip, principal cell, right? CHIEF, chip cell. Chip cell of stomach is responsible for releasing of pepsin, that is initially inactive pepsin, that is called pepsinogen. Chip cell is popularly also chip cell is popularly known as peptic cell, right? Peptic cell, right? It is MCQ point of view, it is very important, right? And as well as short question, and there is also the chance of long question as well, right? And this cell, this beloved cell, is actually considered as CHIEF, chip cell, and this chip cell is responsible for releasing of pepsin, right? Pepsin. And another beloved type of cell which is responsible, which cell, my second question, which cell of stomach is responsible for releasing of HCl? And this HCl producing cell is named as oxyntic cell. Oxyntic cell. And oxyntic cell also popularly known as parietal cell as well. Parietal cell, right? Parietal cell. So first digestion of protein is occur inside the stomach chemically, but our mouth digests the protein physically by the help of teeth, tongue and jaw, right? Now this partially digested protein, now one more important point about you, this protein is now not fully digested, my dear student, focus on that. This stomach protein which is contained by the stomach, now this protein is not yet fully digested under the action of pepsin. That's mean there is some more important enzyme waiting for the digestion of protein, right? This enzyme is really wait for digest this incompletely digest protein. And I want to rub this structure, and after that I need to draw one more diagram here, right? Okay, there is another champion structure, champion organ, another champion beloved organ, right? And this organ is called pancreas. This organ is called what? Pancreas, right? And that is the duct of pancreas, just that is the duct of pancreas, right? And this major duct is finally open into your duodenum, right? This pancreatic duct is directly open into your duodenum, and pancreatic juice, that's mean pancreatic enzyme, protein digestive enzyme, is finally pour into the duodenum, right? That is pancreatic protein digestive enzyme now available in the duodenum. And your food, partially digested food, also come in the duodenum. And this partially digested food, that's mean specially focus on protein, this partially digested protein is now come in contact with this pancreatic protein digestive enzyme. And what happened in this in this duodenum, uh, in the next section we'll learn that, right? Now that is our esophagus, and that is stomach. I just firstly draw this diagram for you again. Sorry my dear. And that is the C-shaped curved duodenum, and your beloved pancreas is situated here, right? That is your pancreas, right? Pancreas, just I roughly draw, and pancreas open into duodenum, and pancreatic juice is just began to flow. This pancreatic juice, pancreatic protein digestive enzyme just began to flow over the partially digested protein, right? This partially digested protein, just consider. Now this pancreas have really a great role. Pancreas have really very champion and master role for the digestion of protein as well as this duodenum part, right? Now you have to focus on the pancreatic enzyme. First of all, pancreas release a set of enzyme for the digestion of protein. First of all, it release trypsinogen, trypsinogen, right? And gen, you know my dear, gen contain, gen is actually the symbol of inactive enzyme, right? You already know that. And that is trypsinogen, which is secreted by pancreas, as well as some another hormone which is also secreted for protein digestion, that is chymotrypsinogen, chymotrypsinogen, and that is also the inactive enzyme. And again another enzyme that is secreted by your beloved pancreas, that is procarboxypeptidase, right? Procarboxypeptidase, peptidase, and pro indicate also the inactive protein. That's mean all this protein which is released from pancreas is initially really a inactive hormone, right? And this hormone is just weeping because of why? Because this hormone is not functional not functional. It is not now not ready for the digestion of protein, right? And now the next idea is that we need to zoom this area. We need to zoom this duodenum part, from where this pancreatic juice, that's mean trypsinogen, chymotrypsinogen, as well as procarboxypeptidase, as well as this fragmented partially digested protein run, right? So this area, right, this area, we need to draw this area in large fashion, right? That is your intestine. And you know very well, intestine contain a special type of cell on it, and this cell is called enterocyte cell. This cell is called, I already mentioned it in carbohydrate digestion, right, and absorption as well, right? This cell is called actually enterocyte cell, which is covered our intestine, right? Enterocyte cell, enterocyte cell, enterocyte cell present in our small intestine. Just simplified way to understand that and realize that, right? This enterocyte cell is our small intestine cell, and this the food is now migrate from this portion, and there is some inactivated hormone that is called your trypsinogen, and there is also some inactivated hormone that is called chymotrypsinogen, as well as some more inactivated hormone which is released from pancreas, that is called, you know very well, that is called procarboxypeptidase. As well as from there is migration of partially digested protein. There is migration of what? Partially digested, fragmentally digested what? Protein. And now the protein is breakdown into dipeptide, tripeptide, and small polypeptide as well, right? And this partially digested protein is now bathed with the inactivated digestive enzyme, and there is a very big role of enterocyte cell for activation of this all of this hormone. In enterocyte cell, actually this enterocyte cell contain specialized enzyme. This enterocyte cell is contain really what? Some specialized what? Enzyme, right? That is specialized enzyme. And this enterocyte cells, this specialized enzyme, name of this specialized enzyme is actually called, this enzyme is called enterokinase. What is this name, my dear? Enterokinase, kinase, right? Now this enterokinase is now sensing, all this hormone, like this first of all this trypsinogen, as well as it's sensing the procarboxypeptidase, as well as it also sensing the chymotrypsinogen as well. But enterokinase enzyme that is present on the membrane of enterocyte of intestine, that specially act on this enzyme is specially, first of all it specially act on fast trypsinogen. When this enterokinase is act on this trypsinogen, then trypsinogen is convert into active enzyme that is called trypsin, trypsin, right? Trypsin. Now this trypsin is going to ready for the digestion of protein, and trypsinogen have the capacity to cut this peptide bond in between the amino acid, right? That is the amino acid This all of this is amino acid, and in between amino acid, this bond is called peptide bond. Peptide bond, right? And in between carbohydrate, this bond is called what? Glycosidic bond, and this unit is obviously will be the monosaccharide, that's mean glucose, right? But here the substrate is actually protein, and protein make a bond between amino acid. This bond is actually called peptide bond, and this active trypsin have the capacity to cut all this peptide bond, and this protein make this protein into a small fragment like amino acid, this that is amino acid and dipeptide as well as tripeptide as well, and some small polypeptide as well. And this trypsin have again very big role for activation of another some enzyme. This trypsin is eventually drop on the chymotrypsinogen, and when on the surface of this chymotrypsinogen trypsin is act, then chymotrypsinogen eventually convert into active enzyme that is called chymotrypsin, that is called what? Chymo, chymotrypsin. Now chymotrypsin is again activated enzyme, and it is ready to digest again the protein, right? And it's may further digest the amino bond between the amino acid, that is peptide bond, and again free the amino acid, more amino acid, again free the more amino acid. And again trypsin also act on procarboxypeptidase, right? And when trypsin act on procarboxypeptidase, then this procarboxypeptidase is again convert into carboxypeptidase, right? This procarboxypeptidase is again convert into carboxypeptidase, right? Carboxypeptidase, peptidase, right? And this carboxypeptidase is again the activated one, right? Activated hormone, and not hormone, that is activated enzyme, and this enzyme again digest the bond between amino acid, that is peptide bond, and there is eventually the digestion of protein, right? After that there is some more, there is lots of amino acid is produced by breaking peptide, polypeptide as well as dipeptide and tripeptide, and make free all of this amino acid. And there is also some peptide present as well there, that is tripeptide, dipeptide as well, right? Tripeptide, dipeptide, and lots of amino acid free by the enzyme trypsin, chymotrypsin and carboxypeptidase under the action of carboxypeptidase. And further action of enzyme is investigate after running this, right? I hope this idea is makes sense for you. And my dear, one more information, till now protein digestion is not complete yet. Till now, just remember, till now protein digestion is not complete yet, right? There is, you can see there is also some dipeptide, that's mean dipeptide is made up of two amino acid, and in between these two amino acid there is one peptide bond, and there is dipeptide, and there is again there is some tripeptide and some small polypeptide as well, right? And you can see, and there is production of lots of amino acid under the action of what? Under the action of chymotrypsin, carboxypeptidase as well as trypsin, right? and further digestion of protein, we are going to discuss that. And now I love to draw this diagram again magnified way in here, right? And again there is the magnified diagram of small intestine, that is duodenum, and further next part of our small intestine. I hope you are understanding me and you sensing this diagram as well, right? That is next part of our intestine, and just consider that is our duodenum, or it may be part of jejunum or ileum, right? And this part is actually contain some specialized cell. I already mentioned that, and this specialized cell is called enterocyte, right? This specialized cell of intestine is called enterocyte. I already mentioned that enterocyte, right? And again enterocyte is responsible for containing a champion enzyme that is called enterocyte, and this enterocyte is responsible for activation of trypsinogen into trypsin, and further trypsin is activated the next pancreatic enzyme, that is chymotrypsinogen into chymotrypsin, and procarboxypeptidase into carboxypeptidase, right? And again, and after the function of all those enzymes, there is production of lots of amino acid, right? There is lots of amino acid free, and there is also presence of some dipeptide, some tripeptide, right? And lots of amino acid as well, right? That is the amino acid, right? Amino acid, and that is dipeptide, that is tripeptide, and lots of that, right? And now this enterocyte cell again contain some more enzyme, right? That is called dipeptidase, right? Dipeptidase. And again there is one more enzyme that is called tripeptidase, right? Just I simplified way I love to express this all this enzyme in that form, right? That is just consider that is dipeptide, that is tripeptide, and already, and one more information, this enterocyte cell also have its own dipeptide and own tripeptide as well, right? And all this enzyme, that is enterokinase and dipeptidase and tripeptidase, all this enzyme is also considered as brush border enzyme, BRUSH, brush, BOARDER, border, brush border enzyme, ENZYME, right? I hope you understanding me. This enterokinase, right? Enterokinase, which is the product of enterocyte, and tripeptidase, dipeptidase is also called brush border enzyme, right? And this brush border enzyme is present on the enterocyte, the cell of small intestine. Now these tripeptidase can attack this bond and again make free the amino acid, and this dipeptidase, not dipeptide, that is dipeptidase, can attack this dipeptide and make free again amino acid. And I also mentioned the enterocyte cell have its own dipeptidase as well as its own tripeptidase as well, right? That is its own dipeptidase, and that is also its own tripeptidase as well, right? And now the actual time for the absorption that is the actual time to understand how our enterocyte absorb these digested protein, that's mean amino acid as well as dipeptide as well as tripeptide, how this enterocyte absorb and eventually it is drained into the blood, right? And you know very well, beneath our intestine there is presence of bloodstream, and bloodstream contain there is lots of blood cell running from this bloodstream. That is actually I eventually draw some RBC, right? RBC, that is our bloodstream, blood, right? That is blood, VLOOD. Now, absorption is nothing but this amino acid is eventually drained into this blood, right? And there is also some ions present inside our intestinal lumen. That is, you know, my dear, that is the intestinal lumen, that is the lumen, and from where the food is moved towards our large intestine and eventually up to the anus, up to the colon, right? And there is some dietary hydrogen ion present, right? Hydrogen ion, not dietary, it is hydrogen ion, as well as there is presence of Na plus ion, sodium ion, right? H plus ion, sodium ion, right? Sodium ion, again sodium ion, lots of this ion is also present. Now, one more interesting thing about absorption, right? There is some co-transporter present on the surface of this enterocyte cell. There is some what? Co-transporter, and this co-transporter have the capacity to take one hydrogen in, and with this hydrogen there is also transport of, actually tripeptide as well, tripeptide as well, tripeptide, right? There is eventually there is presence of tripeptide, right? And this process of absorption is called actually co-transport with the help of hydrogen ion, right? And one hydrogen ion in, and with this one hydrogen there is also the drag of this tripeptide. And you know very well, cell have its own dipeptidase as well as tripeptidase, and that is, you know very well, that is the tripeptidase. And this tripeptidase now act inside the enterocyte cell, and this amino acid make free by breaking this peptide bond in between this tripeptide, and there is real absorption of amino acid. And again there is another transporter. This transporter also transport hydrogen in, as well as it transport dipeptide inside the cell as well, right? There is dipeptidase, and again this dipeptide, not dipeptidase, and this dipeptide is again digested by cells own dipeptidase and make free the amino acid again, right? Make free the amino acid again. And when lots of hydrogen come inside the cell, then cellular pH become low, right? One more understanding point for you: when lots of hydrogen come inside the enterocyte cell, like such a manner, then cellular own pH, mean potential hydrogen is become very very high, and this high amount of hydrogen is leads to low pH, and the low pH is leads to acidic environment in cell, and that's mean there is need to transport back this hydrogen into the lumen, right? And this mechanism, this back transport mechanism, there is another channel, this channel is eventually transport one sodium in and back this hydrogen outside the cell, right? And now your amino acid is inside the enterocyte cell as well, right? And there is another absorption mechanism, that's you consider that is also a transporter of amino acid. This transporter actually transport one sodium in and directly amino acid inside the enterocyte cell. That is the mechanism of direct absorption of amino acid. Direct absorption of amino acid is controlled by the Na plus ion. When one Na plus, one sodium ion come inside the enterocyte, then with this one Na plus ion, one sodium ion, one amino acid come inside the enterocyte cell. And there is the absorption mechanism of tripeptide and dipeptide with the help of sodium co-transporter, right? This transporter is called sodium co-transporter. Now this enterocyte contain lots of amino acid inside this, and this amino acid now need to transport in blood, exactly, right? Now there is no special mechanism to absorb this amino acid into the blood. That is actually directly transport by the diffusion, by the simple diffusion. That's mean it is active transport mechanism. By the active transport mechanism, this amino acid is transported, directly in the bloodstream, right? Directly into the bloodstream. Actually that is called active transport, and active transport is responsible for breakdown of ATP into ADP. So there is a ATP breakdown. By expending energy, this amino acid is transported, absorbed eventually into the blood, right? And that's why it is called active transport, because there is expenditure of energy, right? And one more point to understand that, so there is amino acid, and this amino acid is absorbed into the blood by the diffusion process, again I mention, right? This amino acid is absorbed in the blood by the diffusion, simple diffusion as well as active transport as well, right? Now there is another point, right? There is another transporter, that is called sodium transporter. There is also sodium present in the enterocyte cell. This sodium can enter into the bloodstream, right? And this sodium can help this amino acid to transport inside the blood as well, right? That is another mechanism of digestion, absorption of protein, right? When this Na plus is come inside the blood, then one potassium ion is exchanged into the cell by the transporter. That is the whole idea about the amino acid digestion and absorption as well. Now this amino acid is transported into the hungry cell, and after that the excess amino acid is stored into the liver. That is the whole peaceful idea of digestion and absorption of protein. I hope you enjoy a lot this video and it makes sense clear. Thank you for watching. Like, comment, share and provide feedback. Thank you. Bye. Sorry, my dear, there is a small correction. Actually, sodium, there is no sodium ion activity to absorb the amino acid into the blood directly, right? A small correction there, right? Just 1 minute. There is a small correction, right? Right. Okay. Now, all this amino acid is directly transport by the diffusion process from this enterocyte cell to blood, right? And again, there is we need to understand that there is amino acid, and this amino acid, just consider this amino acid again enter into the blood, and there is another exchanger. There is some sodium potassium exchanger, right? There is some sodium potassium exchanger. This sodium potassium exchanger do that, the one sodium come inside the blood and one potassium go back to the enterocyte. That is the correction, right? There is no direct absorption of amino acid with the help of sodium into the blood, right? Actually, there is sodium potassium exchanger, and when one sodium come inside the blood from this enterocyte, then one potassium go into the enterocyte cell from blood, and there is exchange, simply diffusion, exchange of amino acid by the process of simple diffusion or active transport. That is the whole idea of your beloved topic, that is carbohydrate, not protein digestion as well as protein absorption. Thank you. Bye, bachcho.